Introduction. Biochemistry plays a pivotal role in understanding the intricate processes of human metabolism. One of the fundamental aspects of metabolism is glucose homeostasis, which is tightly regulated in order to maintain optimal energy levels for the body. Gluconeogenesis, the process by which glucose is synthesized from non-carbohydrate precursors, is a critical pathway that ensures glucose availability during periods of fasting, starvation, or increased energy demand. Understanding the regulation and intricacies of gluconeogenesis is essential for comprehending various metabolic disorders and their implications in human health. This script aims to provide a comprehensive understanding of gluconeogenesis and its regulation, as well as other related topics that contribute to glucose homeostasis. Gluconeogenesis is regulated by a complex interplay of hormonal, enzymatic, and transcriptional mechanisms. The key enzyme controlling the initiation of gluconeogenesis is phosphoenol pyruvate carboxykinase, PEPK, which catalyzes the conversion of oxaloacetate to phosphoenol pyruvate. PEPK activity is regulated by several factors, including glucagon and cortisol, which stimulate its expression. Additionally, the ratio of NAD/NAD+ plays a crucial role in regulating gluconeogenesis, as it determines the activity of enzymes such as malate dehydrogenase and pyruvate carboxylase. The precise control of gluconeogenesis ensures glucose production is modulated according to the body's needs. Glucose 6-phosphatase deficiency, also known as von Gurke's disease, is a rare genetic disorder characterized by the impaired conversion of glucose 6-phosphate to glucose. This deficiency leads to glycogen accumulation in the liver and kidneys, resulting in hepatomegaly and growth retardation. Glucose 6-phosphatase plays a crucial role in the final step of both gluconeogenesis and glycogenolysis, making this disorder detrimental to glucose homeostasis. Patients with glucose 6-phosphatase deficiency require careful dietary management and may benefit from enzyme replacement therapy. Glycogen branching enzyme, GBE, is responsible for the formation of branches in the glycogen molecule. Through its activity, GBE generates more non-reducing ends, allowing for efficient glycogen degradation. Deficiencies in GBE result in glycogen storage diseases, such as Anderson's disease, characterized by abnormal glycogen structure and impaired glycogenolysis. Understanding GBE's role in glycogen metabolism provides crucial insights into glycogen-related disorders and their impact on glucose homeostasis. Fructose metabolism differs from glucose metabolism, and its unique processing in the liver contributes to overall glucose homeostasis. Upon ingestion, fructose is rapidly metabolized into fructose 1-phosphate by fructokinase. Fructose 1-phosphate is then cleaved into glyceraldehyde and dihydroxyacetone phosphate, DHAP, by aldolase B-DAP can either enter glycolysis or gluconeogenesis pathways. The regulation of fructose metabolism is critical in maintaining hepatic glucose production and preventing excessive triglyceride synthesis, which is closely linked to metabolic disorders such as non-alcoholic fatty liver disease, NAFLD. Glycogen synthase kinase 3, GSK3, is a key regulatory enzyme involved in the control of glycogen metabolism. GSK3 phosphorylates and inhibits glycogen synthase, thereby decreasing glycogen synthesis. Insulin, through its signaling pathway, inhibits GSK3, promoting glycogen synthesis and storage. Dysregulation of GSK3 activity has been implicated in conditions such as insulin resistance, type 2 diabetes, and neurodegenerative disorders. Pyruvate carboxylase is an essential enzyme involved in gluconeogenesis. It catalyzes the carboxylation of pyruvate to oxaloacetate, a precursor required for glucose synthesis. The regulation of pyruvate carboxylase activity is tightly linked to metabolic conditions and hormonal signaling. Under fasting or high energy demand states, pyruvate carboxylase is upregulated to provide the necessary substrates for gluconeogenesis. Understanding the role of pyruvate carboxylase sheds light on the metabolic adaptations necessary for glucose production under various physiological conditions. Hepatic insulin resistance is a hallmark of type 2 diabetes, characterized by impaired insulin signaling and glucose intolerance. Insulin resistance in the liver disrupts the suppression of gluconeogenesis, leading to increased hepatic glucose production. Dysregulation of various signaling pathways, such as those involving insulin receptor substrate, IRS, proteins and protein kinase B, ACT, contribute to the pathogenesis of hepatic insulin resistance. Elucidating the mechanisms underlying hepatic insulin resistance is vital for developing targeted therapeutic interventions for type 2 diabetes. During periods of prolonged fasting or starvation, gluconeogenesis becomes the primary supplier of glucose to maintain vital functions. The regulation of key enzymes, including PEPK, is enhanced, while glycolysis is downregulated to conserve glucose. Hormonal signals, such as glucagon and cortisol, play a pivotal role in orchestrating the metabolic shifts required for efficient gluconeogenesis. Understanding the adaptations that occur during fasting states provides valuable insights into the metabolic resilience of the body and the challenges associated with prolonged energy deprivation. Glucagon-like peptide 1, GLP-1, is an incretin hormone that exerts several important effects on glucose homeostasis, including modulation of hepatic glucose production. GLP-1 promotes glucose uptake by hepatocytes, inhibits glucagon secretion, and suppresses hepatic gluconeogenesis. GLP-1-based therapies, such as GLP-1 receptor agonists, have emerged as effective treatments for type 2 diabetes. Understanding GLP-1's role in hepatic glucose metabolism provides insights into its therapeutic potential and its impact on overall glucose homeostasis. Hexokinases are critical enzymes involved in the phosphorylation of glucose, a key step in both glycolysis and gluconeogenesis. In the liver, different hexokinase isoforms exist, each with unique regulatory properties. Hexokinase 4, glucokinase, is predominantly expressed in the liver and displays low affinity for glucose. This allows the liver to efficiently remove excess glucose following meals and acts as a glucose sensor for insulin release. The intricate regulation of hexokinase isoforms ensures precise control over hepatic glucose metabolism and influences glucose homeostasis. The Cori cycle describes the reciprocal relationship between gluconeogenesis in the liver and lactate production in peripheral tissues, particularly skeletal muscle. During intense exercise, skeletal muscle produces lactate, which is released into the bloodstream and transported to the liver. In the liver, lactate is converted to pyruvate, which serves as a substrate for gluconeogenesis, ultimately leading to glucose production. The Cori cycle plays a crucial role in replenishing glucose stores and maintaining energy balance during periods of high energy expenditure. Galactose, a component of lactose, has its own metabolic pathway in the liver. Galactose metabolism involves several enzymatic steps, including its conversion to glucose 1-phosphate by galactose 1-phosphate gyrosyltransferase. Inherited deficiencies in galactose metabolism enzymes result in galactosemia, a disorder associated with potentially severe liver complications. The understanding of galactose metabolism provides insights into the liver's ability to handle specific substrates and highlights the consequences of metabolic disruptions in this pathway. The intricate regulation of gluconeogenesis and related processes is essential for maintaining glucose homeostasis. Pathways and enzymes involved in glucose metabolism play critical roles in various metabolic disorders, necessitating a comprehensive understanding of their underlying mechanisms. The topics covered in this script touch upon different aspects of glucose homeostasis, offering insights into the complex interplay of hormonal, enzymatic, and transcriptional regulation. By delving into these intricate pathways, we gain a deeper appreciation for the remarkable biochemical processes that enable the human body to maintain optimal energy levels and overcome metabolic challenges.